Evening and welcome to the 2021 Koch Institute for Integrative Cancer Research Image Awards event. I'm Daryl Irvin, one of the Associate Directors of the Koch Institute. Tonight is the first of many events planned to celebrate our anniversary year, and it's a fitting one. First, this date is special because exactly 10 years ago today, we dedicated the Koch Institute Research Facility, MIT's Building 76. Through its careful design, Building 76 represented not only a major improvement over our previous space, but it created an opportunity to build a new community around our collaborative cancer research model, bringing scientists and engineers together under one roof to solve the toughest problems in cancer biology and develop new treatment and diagnostic solutions. This broad community draws on many different departments at MIT, engages collaborations at nearby institutions, hospitals, and companies. It involves alumni and the public in the research enterprise through our public galleries, where tonight's images hang, and programs like the presentations we're about to hear. Second, the first thing people see when they come to visit the Koch Institute are the exhibits in our public galleries, specifically the 10 8-foot tall image installations showcasing life science and biomedical research from MIT labs. It's part of MIT's commitment to make our research accessible to everyone. If we can't bring you into the labs, and that's even more true this year than usual, then we can bring the research to you. These images also represent the past decade of progress in fighting cancer, uncovering key insights into the way cells work, and pushing the envelope of what is technologically possible. Tonight, you'll hear from people who are studying the inner workings of cells, developing new platforms for cancer diagnosis and treatment, and ensuring that science, technology, and healthcare work for everyone. In that spirit, we'll also revisit an image from our very first Image Awards exhibition and hear from the project PI about the extraordinary advances her lab has made on this work over the last 10 years. As I said, this is the first celebration of many. Our master anniversary events calendar will be released soon, and we sincerely hope you'll join us for more webinars throughout the year, perhaps even some in-person programs as circumstances allow, and help us celebrate the people, projects, and promise of integrative cancer research. With that, I welcome you to the 2021 Image Awards, and I'll turn things over to Erica Reinfeld, your MC for the evening.
Thank you so much, Daryl. I am so thrilled to be showing off this year's images and celebrating the stories behind them. I'm not going to wax poetic because we have a lot of territory to cover this evening, but I did want to thank everyone who has submitted images over the years, and especially this year during the pandemic, as well as the judges who helped us curate this year's beautiful exhibition. As Daryl said, we'll be hearing about 10 different images tonight. Everyone, each team will take the stage for 5 minutes or less, and then they'll take questions using the Zoom Q&A interface at the bottom of the screen. So if you want to know more at any point, just type in your questions and the presenters will do their best to answer them in writing post lightning talk. So we're going to start things off with a project from the Batia Lab. This is one that touches on many of the Koch Institute's official research focus areas, and even more importantly, some of the key challenges in cancer research, how to find tumors early, understand their evolution, and eventually stop them in their tracks. So it's a tall order, but let's cut right to it.
Yes, okay, so I'll just start off again. So as I mentioned, I'm Ava, and together with Jesse and on behalf of the Batia Lab, I'm so excited to tell you more about our image, Making the Cut, probing enzyme activity with nanosensors. As you know, cancer is characterized by the uncontrolled growth of tumor cells that spread and invade the tissues in the body. But how are cancer cells actually able to do this and invade and spread in this way? It turns out that cancer cells produce a special type of protein, an enzyme that's called a protease, which I'm showing here as these blue Pac-Men. And what these proteases do is they actually can break down the tissue surrounding the cancer cells, allowing them to spread and invade. These protease enzymes are able to break down the tissue in this way because they have this very special property, in that they're actually able to recognize other proteins, which I'm showing here with this red curve, and cut them, breaking them down. In the Batia Lab, we're very interested in understanding more about what these proteases are doing and how they contribute to cancer. To do that, we used engineering methods to design nanosensors that can measure how and where these protease enzymes are active. The nanosensors we build are designed to be recognized and cut by proteases. And we've designed them in a special way such that a part of the sensor that's released by cutting can actually bind tissue at the precise spot where that cutting happened, such that we can visualize protease activity using microscopy. When we test these sensors on slices of tissue, like from a patient's tumor biopsy, we can visualize and localize where these cutting events happen in order to understand where these protease enzymes were active.
So Ava has introduced you a probe that allows us to visualize where these certain enzymes called proteases localize in a tissue. And so our ultimate goal was to apply them to cancer and to learn how proteases, the role that proteases play in cancer. But before we applied this approach to cancer, we first wanted to test it in a simpler and more well-defined tissue. So here I'm showing you a zoomed in section of the colon, which we sliced and mounted on a microscope slide. So each blue dot that you see here represents one cell nucleus, which you can see are all over the colon because all of the cells in the colon have a nucleus. The cells labeled in green are called epithelial cells, and these are the cells that make up the lining of the colon. These cells are known to produce large quantities of proteases, which are thought to help the body maintain a healthy relationship with the trillions and trillions of bacteria in the gut. So when we applied our protease activity nanosensors to this colon slice, we expected to see the green cells light up because these are the cells that come into contact with those gut bacteria. And this is exactly what we saw. So as you can see here, the magenta signal corresponding to protease activity is illuminating the green epithelial cells. So these findings were extremely exciting because they proved to us that our protease nanosensors can be used to visualize protease activity in a tissue, and suggested to us that we could maybe apply the very same approach to many different diseases, including cancer. So since we captured this image, we have done exactly that, which has allowed us to shed light on the many important roles that proteases play in multiple different malignancies, including prostate and lung cancer. And our lab is now working on using this information to target protease activity in order to enable earlier detection and treatment of cancer. So with that, we will just conclude and thank the people who made this work possible. In particular, our advisor, Dr. Sangeeta Bhatia, who has encouraged us to take risks and get creative with our science. And thank you also to the Koch Institute and our generous funders for supporting this work. And finally, thank you all for joining us here tonight.
Thank you, Jesse and Ava. Thank you so much. Those of you out in the audience, please be liberal in putting questions into the Q&A. They will check those out and do their best to answer them. And now we are going to head over to another multidimensional platform. So this next project builds on this idea of tools that can do multiple functions. This particular tool takes on diagnosis and drug delivery all in a single device. So this image was taken at one of our faculty spin-out companies, but the science and technology is pure MIT.
Hi everyone, I am Nuria and I am a PhD candidate at the RT Lab where we integrate tissue-responsive biomaterials into the medicine field. And today I am very honored to present one of these projects to you. So over the years, hypodermic needles have been the preferred device to administer drugs. One wonders why, since they are invasive, painful, they need to be handled by medical professionals, and let's be honest, who isn't afraid of them? For this reason, scientists, we have been working to develop potential replacements for these scary devices, and our best shots so far have been microneedles. Microneedles are a medical device that contains hundreds of microscopic needles capable of piercing the most outer layers of the skin. Microneedles can be self-administered, like in a Band-Aid, and they are not painful because they do not reach nervous terminations. Microneedles were originally designed to deliver different types of drugs, but in recent years, microneedles have also been proposed as a diagnostic tool because they can extract interstitial fluid. Interstitial fluid is a tissue that is very rich in immune cells and other soluble biomarkers. So if we sample it, it can give us information on the patient's health, just like a blood test. Inspired by the research performed at the Institute, our group has developed a microneedle platform that integrates a therapeutic and a diagnostic arm within the same patch to treat cancer. Specifically, we delivered an immunostimulatory drug that awaits the fighter cells of the immune system to fight the tumoral ones. Simultaneously, the immune cells that surround the tumors can penetrate inside the microneedles. So once we recover the microneedle patch, we can analyze the composition of the interstitial fluid that has infiltrated. This platform works as a non-invasive biopsy. So depending on the cell populations and biomarkers that we identify, we can know whether our drug is working and if we indeed awake the immune system to fight the tumors. So we took our winning picture during one of the first experiments of this project, when we were struggling to design a new and unique biomaterial that could be used to fabricate the microneedles. For us, this was the challenge of the project, because the microneedles had to be strong enough to penetrate the skin and release the therapy, but at the same time, they had to provide this nice and cozy place for cells to flow inside. To meet this diagnostic goal, we had to design microneedles that could be entirely disintegrated, but only after retrieving them from the patient. Once out of the body, we would dissolve them and analyze their contents. Engineering the microneedles so they would have this on-demand degradation feature was really challenging because we had to ensure that the disintegration process wouldn't damage the cells. Otherwise, this could have biased our readout. Finally, we had our eureka moment, and we identified a top polymer. We included in the structure of this biomaterial what could be defined as a chemical switch. So when we turn it on with a reagent, we can dissolve all the microneedle in less than 5 minutes and analyze the different cellular populations and soluble biomarkers that are entrapped inside. We confirmed that our therapy was really effective because we saw a shift in the balance of fighter cells versus the disarmed ones. Our results confirmed that the number of fighter cells increased, and this was accompanied by a dramatic shrinkage of the tumor size. We are very excited because this device could be a revolutionary tool for clinicians, since these microneedles can deliver a therapy against cancer in a non-painful way and simultaneously provide a readout in real time of the patient's state to inform on whether the therapy is working before the disease reaches an irreversible state. Last but not least, I'd like to thank my advisor, Professor Arzi, and all my colleagues and friends from the lab for all the support and help during this fast month. Thank you, everyone, and thank you all who joined tonight.
Thank you, Nuria. And I know you've called in from Spain, so thank you so much for. I think you win the award for distance not traveled on this presentation. And I see we've already got a question come in for you on the Q&A, so keep those coming, and we'll move along to the next image. You mentioned immune cells, which is exactly the breadcrumb I need to move us along to our trash talk, which, despite the name, is actually a very supportive collaboration between cancer researchers at the Koch Institute and neuroscientists at the Picower Institute for Learning and Memory right across the street. So Nader, you are up.
Hi, everyone. My name is Nader Morshed, and I'm excited to talk to you today about the project that I worked on in Forrest White and Li Wei Tsai's lab. And the goal of this project was to perform a signaling analysis of Alzheimer's disease in order to better understand which signaling networks were changing in the disease in order to better develop therapeutics against Alzheimer's disease. And to begin with a bit of background, Alzheimer's disease is the most common form of dementia that's characterized by neurodegeneration in regions of the brain that are associated with memory and cognition. And the brains of Alzheimer's patients have been characterized as having these two key pathologies: amyloid beta plaques which are composed of short protein fragments that bind together into large aggregates, as well as neurofibrillary tangles, which are composed of a protein called tau, which is phosphorylated in disease. And this phosphorylated tau binds together into large aggregates within the neurons and contributes to their degeneration. However, there's a hidden player that's not visible in this image here, and that is the microglia, shown here in green. The microglia are the brain-resident immune cells that play an important role in keeping the brain free of foreign pathogens. But these microglia have a complex role in the brain and contribute to learning and memory by forming many tight junctions with the neuronal synapses, as well as the neuronal bodies. And they signal back and forth to these neurons in order to mediate learning in the brain. And in disease, microglia have an important role in breaking down protein aggregates. And the reason why we became interested in these cells was a result of a phosphoproteomics analysis that we performed for our project. Phosphoproteomics is a technique that allows us to take a brain tissue dissection, and in our case, we used brain tissue from several different mouse models of disease. And this technique allows us to enrich for the phosphorylated sites on proteins and then analyze the abundance of these phosphorylation sites using mass spectrometry. And what we found in our analysis was that there was this protein, Siglec-F, that had increased phosphorylation across all of the different mouse models and brain regions that we looked at. And to give a little bit of background on the Siglecs, these are a family of immunomodulatory receptors that bind to sialic acid, which is a glycan, and alter the cellular signaling immune response of the cell that is expressing this receptor. And so Siglecs have an important role in recognizing the sialic acid pattern present on cells and allows an immune cell to distinguish host cells from foreign pathogens. However, these Siglecs could have a pathogenic role in disease. And so we hypothesized that this receptor was expressed in the microglia in the brain. And so that led us to take this image, which is up in the gallery today, showing the microglia in magenta, expressing Siglec-F in cyan, in close proximity to an amyloid beta plaque, shown in yellow. And you can see that the Siglec receptor forms a tight surface around the dense core of the plaque, suggesting that it may be altering the microglia's ability to respond to this protein aggregate. And so going forward, we'd like to better understand what is the functional role of Siglecs on microglia in diseases of the brain. But I'll cut it short here and just thank the people who supported this work. The White Lab and SciLab are full of a bunch of amazing and wonderful people, and especially Forrest White and Li Wei Tsai are great mentors who taught me a lot. And I'd especially like to thank Alexi Knott and William Ravenius, without whom this work would not have been possible. And I'd also like the institutes. I'd like to thank the institutes and funding sources who supported this work. And if you have any questions, just ask them in the chat. Thank you for your time.
Thank you, Nader. That was great. And it was a great introduction to talking about cell function and what's happening between cells, inside and outside cells, because our next presentation, we're actually going to dive deep into the heart of a cell. And we're going to talk again about what happens to cells over time. So now Anya is going to take you through the ages.
Hello, thank you for the opportunity to give you a glimpse of my work. My name is Anya, and I'm a postdoc in the Sabatini lab. And I'm gonna take you through the ages and tell you about our exploration of mitochondrial structure and function. The image that is in the gallery was taken in collaboration with Peggy Bisher and David Mankus from the NanoCore at the Koch Institute. So we live in a rapidly aging world, And across the globe, you can see that the average age of societies have been increasing. This has been happening over time, and it's projected that this tendency is gonna continue in the future, so that in 2050, we will have 2 billion people who are aged 65 and over. And this produces a number of challenges for societies across the globe. For example, aging is one of the factors, risk factors for many age-related diseases. And so there's a lot of interest in the biomedical community in understanding the causes and mechanisms that underlie aging, with the hope that in the future we will be able to better address diseases of old age. There's a number of molecular features of aging cells that are quite distinct and different from those of young cells. And these vary from differences in communication between cells, the regulated nutrient sensing, or differences in genomic stability. But one interesting fact is also that the compartments within cells age as well. And the mitochondrium is one of the organelles that is known to age and undergo most dramatic changes. So what are mitochondria? Mitochondria are compartments within cells, and here they're shown in red. And you may remember that they are the powerhouses of the cell because they provide energy for lots of different activities that cells perform. However, mitochondria are much more than that. They're important for a number of different metabolic pathways. They are key hubs for cellular signaling, and they can even decide cellular fate. As I said, they are one of the organelles that are known to undergo very dramatic changes in aging. And while there is lots of very interesting and beautiful literature out there on characteristics of aged mitochondria, what has been missing is a very comprehensive and unbiased analysis of aged and young mitochondria. And this has not been possible before because we have lacked tools to do such a big analysis. However, in our lab, we have developed new biochemical approaches that allow us to rapidly isolate mitochondria from young and aged tissues. And these approaches are compatible with a number of downstream analysis platforms, which allow us to profile metabolites and proteins and really zoom into very detailed changes in composition of mitochondria as we age. We hope to combine this biochemical analysis with powerful imaging approaches to look at structure, ultrastructure, numbers, and sizes of mitochondria through the ages. And this image represents one of the images that we have taken across the tissues and through the ages. It is an image taken in the liver. And each of these colored blocks represents a single separate cell. You can see within each of the cell, you can see these membrane-bound compartments, and these are the mitochondria. And we are currently performing analyses of the young and aged mitochondria in different tissues and comparing their size, structure, and number. And we hope to combine this with our biochemical analysis to form novel hypotheses about what happens in aging. And with that, I would like to thank David Sabatini, who is the fearless leader in our lab, for his support and encouragement to work on this project. I would like to thank the Peterson Nanotechnology Materials Core Facility, Peggy, David, and Abigail, without whom the electron microscopy image and the whole series of the images we've taken would never be possible. And I would like to thank all the members of the Sabatini lab, as well as the Metabolomics Core, for creating a wonderful environment to work and do science. And thank you all for listening.
Thank you so much, Anya. So I do want to note that we have David and Peggy behind the scenes as well, so they can see your questions. So if folks have questions about the microscopy there, they'll be happy to jump in and answer. I see that there's activity going on in the Q&A, which is great. Thank you everyone who's asking and answering questions. As Anya mentioned, Mitochondria are responsible for much of the metabolic functions of the cell, which gives us a beautiful transition over to the Yilmaz lab, where Shinya is going to talk to you about stem cell regeneration in the intestines.
Okay, thank you for the introduction, Erika. Hi, everyone. My name is Shinya Imada. So I'm a postdoc at Wilmot Labs. So I'm really happy to share this moment with my colleagues because this award wasn't just thanks to one person, but to my colleagues' effort. Okay, so the left figure shows the structure of small intestine. The inner lining of the gut consists of the single layer of intestinal epithelium, and they play the important role for the nutrition absorption and prevention of the pathogen invasion. The intestinal epithelium is composed of villus and U-shaped crypt parts, and at the crypt bottom there are stem cells. So right figure shows the epithelial regeneration. The stem cell existing at the crypt bottom give rise to the transit amplifying T cells. And T cells generate the differentiated cells. And these differentiated cells migrate to the tip of villi and expel into the intestinal lumen. So most of the epithelial cells generated from stem cells are replaced every 3 to 5 days. So that in small intestine, there are Paneth cells adjacent to the stem cell. They support the stem cell maintenance. Moreover, in the tumor case, stem cells are considered to be the cell of origin. So in this way, the stem cells are a key player for the intestinal physiology and cancer. So for this reason, we are studying the intestinal stem cell biology. So Wilmot's lab is especially interested in the interaction between the diet and the stem cells. And we are studying the regenerative activity of stem cells under different dietary conditions, such as the high-fat diet and intermittent fasting. So intermittent fasting intervention is well known to have a lot of benefit for our health. So and it has two phases. That is the fasting phase and the refeeding phase. So I'm very interested in the stem cell response to the different phase and studying the stem cell function in each phase. So to address the stem cell function under different dietary conditions, so we are using the various genetic mouse model. So this picture mouse model enables the red fluorescent labeling of stem cells and progenies derived from stem cells upon the region administration. So this yellow arrow, the stem cell, which have thin shape and red fluorescent. This asterisk shows the Paneth cells marked in green. And this pink dot allows show the progeny, which are newly generated from stem cells post-region administration. So you can see there are still non-red fluorescent epithelials marked in blue at the upper crypt, because it takes 5 days for all epithelial replacement, and we correct this intestinal tissue 2 days post-region administration. And this figure shows the comparative experiment result using this model. When we label the fluorescent in the stem cell from control and post-fast refeed mice, the number of the progeny generated from stem cells in a certain period is larger in the refeed mouse compared to the control mice, indicating that the post-fasting refeeding state increase the regenerative capacity of the stem cell. So from this result, I'm also studying the how post-fast refeeding impact intestinal stem cell intestinal tumor initiation. Okay, I'd like to thank my mentor, Professor Mel Wilmot, offering the guidance and suggestion for my project. Lena and Corina, So he was a visiting student from Brazil. He helped my project a lot. So I'm grateful for his effort and support. And I also would like to thank Chao-Wei Cheng for establishing this genetic mouse model. I also appreciate Lab Manager Kerry. So these are research found, and this is my fellowships. So lastly, the picture was taken almost 2 years ago with my kids. So this year, I hope to share my image with them. Thank you for your attention.
Thank you, Shinya. I hope so too. I hope everyone gets a chance to visit the facility and see these images in person. But we are recording this presentation and it will be online so that people can watch it after the fact. All right, so we've got one more trip into one more deep dive into cell biology. And for that, we're going to take a dip in the nucleotide pool.
Thanks, Erica. Yeah, my name is Luke. I'm going to be talking to you today about my image I'm calling Nucleotide Pool, Understanding Disruptions in Cell Division. So one of the primary goals of biology is to understand how cells function and how their functions are regulated. And one window into this is looking at the cell's genome. The genome is contained in each cell inside the nucleus. And for humans, the genome contains about 3 billion base pairs of DNA. These are the A, G, T, C letters you think of when you think of DNA. These base pairs are organized into functional units called genes. And about 20,000 of these genes are what we call protein coding genes. And these genes provide specific instructions for making proteins. And proteins are the main workhorses for the major functions of cells. However, for many proteins, the full range of how they work inside cells is not really well known. So when we're studying a process of interest, we often ask what proteins are involved. The process I'm going to talk about today is mitosis. It's a basic process of cell biology of cells duplicating their genome and splitting into 2 subsequent cells. It's really important for a lot of different processes in biology, and especially for some pathological conditions, including cancer, for cells to be able to regulate how they progress through mitosis. So one way to study the functions of genes and their protein products is by disrupting or perturbing genes of interest. Here I'm demonstrating that by turning part of the genome red. In modern biology, this is usually done using a CRISPR-Cas9 system. After perturbing a gene, we want to know how that affects the cell's function or its phenotype. So we could measure a variety of different things, including cell shape, size, how it progresses through mitosis, responds to stimuli, or any other thing that we're interested in. When we're studying a process and want to know what genes are involved, we often will do a large screen where we perturb many different genes and see which ones affect the process that we're studying. Traditionally, this is done using arrayed screening, where we use multi-well plates like the one shown here, where each of these little pink circles represents one well where a population of cells is growing and one gene in each of these individual wells is perturbed. This is nice because you know which cells had which perturbation, how each cell was disrupted. You can make all kinds of different measurements, including using microscopy, measuring gene expression changes, etc. The downside for arrayed screening is that it's difficult to scale and often requires laboratory automation to test large lists of genes. More recently, biologists have started to use what's called pooled screening. In this case, you have a large population of cells when each cell has a different gene perturbed. In this way, you can test thousands of genes in a single sample or a single pool of cells. However, traditionally, these sorts of pooled screens are limited to growth phenotypes. Here I'm showing a brief overview of a traditional pipeline of pooled cell screening. In this case, the phenotypes that you can measure have to be linked to how fast cells grow in this specific condition. In addition, the measurements that you're making are only averages across populations of cells. On the bottom, I'm showing a new technique we've developed in the Blaney lab where I work. In this case, the cell phenotypes are measured using microscopy, and after measuring the cell phenotypes, we use a process called in situ sequencing, where we sequence in place in each cell a DNA barcode that tells us which gene was perturbed in each individual cell. The advantages for this approach is that one, you can measure a wide range of different phenotypes, not just growth phenotypes. And also you get single cell information, so you know how each individual cell responded to the perturbation. Here I'm showing an image one out of thousands of images from an optical pooled screening experiment I recently did. Here in magenta, I've shown the nucleus of each cell, and green is a stain for tubulin, which is a structural protein involved in mitosis. So what we can do when we have lots of these images of individual cells, we can identify cells with phenotypes of interest. Here I'm highlighting 4 cells that happen to be in mitosis when I fix them, of varying structures, some more normal, like in the bottom left, and some much more perturbed. However, we don't know from this image what gene was perturbed and why that causes cell to not progress through mitosis normally. This is where the in situ sequencing data comes in. So in this image, each of these individual colored dots represents one read of the in situ sequencing data. And the color of the dot tells us which DNA base was in that position for the DNA barcode. And if we iterate through multiple cycles of sequencing, we can build up a short DNA barcode sequence that can then tell us, based off our experiment design, what gene was perturbed in each of these individual cells. Here I'm showing a split view. On the left is the phenotype information, so the DNA and the tubulin stains. On the right is the in situ sequencing information for the same set of cells, showing us which gene was perturbed in each of these individual cells. This is the image that's hung in the Koch gallery, except for not with the animations. With that, I'd like to thank the Blaney lab where I work, and especially my advisor, Paul, and then in addition, Avatar, David, and Becca, who originally developed the optical pooled screening approach. And this mitosis project in particular has been a close collaboration with Ian Cheeseman and Kwan-Chung Su in their lab at the Whitehead Institute.
Thank you, Luke. And again, please direct your questions to the Q&A. Our panelists are on the lookout. All right, so all of that talk of mitosis makes me think that maybe we should grow some cells. And so for that, I'm going to call Arnav up to talk about his image, and be careful on this one. You got to mind the zap.
Awesome. Thank you for the kind introduction, Erica. It's an honor to be presenting at the KI Image Award ceremony. So my name is Arnav Chhabra, and I'm an alum of the Koch Institute. I actually used to be a postdoctoral fellow in Dr. Sangeeta Bhatia's lab there. So at the Bhatia lab, we're a team of engineers who like to derive inspiration from biology. As an example, we'll take a novel scientific finding and leverage that discovery to develop a tool that will either help us with early detection of disease or help us improve an actual therapy. The clinical problem that really motivates us is highlighted in this graph here. For people with end-stage organ failure, their only option is a transplant. However, the supply of these organs is very limited. As you can see, the organ wait list has rapidly increased over the last 20 years, while the number of transplants and donors has not kept up. To bridge this gap, many researchers, including our lab, is utilizing cells as therapeutics. But cell therapies suffer from one major problem. They need blood vessels to function well when they're implanted in the body. The blood vessels represent the highway that transports nutrients and oxygen to all of its cells. In these images, I'm showing what happens when you don't have a blood vessel in a mass of tissue. As you can see in the left image, the cells in the center start to die due to a lack of nutrients and oxygen. While there are many different ways to think about this problem, we actually look towards cancer to inspire us. So cancer cells are incredibly pesky. As they grow and invade your body, they produce these signals to recruit blood vessels to help sustain themselves. So we thought to ourselves, can we leverage the same mechanisms, but to help form blood vessels instead inside of our cell therapies? We looked at many different ways to do this. And after a lot of research, we actually arrived at a very interesting method. As it turns out, you can electrically stimulate tissues in a dish to help form blood vessels. And by using electrical signals as a stimulus, we are able to form these blood vessels in an extremely controlled way. We can detect areas of the tissue that are low in oxygen and nutrients and stimulate those regions to promote the formation of blood vessels. And the result is as follows. So to create this image, we used electrical fields to fabricate a network of blood vessels, which are shown in the red, around engineered mini livers, which are shown in cyan. The resulting vasculature efficiently transports nutrients to the mini organs, promoting their long-term survival and demonstrating the potential of electrical stimulation to improve both transplant technology and cell-based therapies. We think this approach has a lot of potential to improve cell therapies and in turn improve outcomes for patients suffering from end-stage disease. And with that, I'd like to acknowledge a whole host of people. I'd like to thank my project partners, Dr. Edward Tan, who's a postdoc in the Bhatia lab, and Keval Vyas, who was a technician in the Bhatia lab. I'd also like to thank my entire lab, specifically my PhD and postdoc advisor and longtime mentor, Dr. Sangeeta Bhatia. Lastly, I'd also like to thank Erica for running the Koch Image Awards and the 2021 judges for selecting our work.
Thank you, Arnav. I see what you did with the potential of this project. All right, our next image actually builds on this idea of blood vessels as a transport network. Here they are right on the screen. So Joelle, why don't you jump right in?
Thank you, Erica. I'm going to present here a series of images that we titled Tortuous Trafficking, and these are images of the blood-brain barrier. So taking these images was really a team approach, and I wanted to start off by telling you a little bit about our team. My name is Joelle Strela, and I'm a pediatric neuro-oncologist, so that means I take care of children with tumors of the brain and spinal cord. I also am a researcher, and I'm a postdoctoral fellow in the laboratory of Paula Hammond here at the Koch Institute. The thing that really brings my two jobs together is the desire to have better treatments for brain tumors. In order to do this, I really needed to study the blood-brain barrier. And this is where Jeff comes in, and he's a panelist today and able to answer your questions through the chat. Jeff's an expert in intravital imaging, which is the imaging of live animals at microscopic resolution. We teamed up to do this project in order to study nanoparticle trafficking across the blood-brain barrier. So to do this, I first perform a surgery in an anesthetized mouse, so similar to what happens for a patient surgery, and then make a small window in which we can look into the brain. And then Jeff operates a very precise form of microscopy called two-photon microscopy in order to take these time-lapse images that you're seeing a screenshot of when we do this. So it probably needs little introduction to this group, but brain tumors are exceptionally hard to cure. And you've probably heard of at least one of the most common types of brain tumors called glioblastoma multiforme. Unfortunately, we've made very little success in curing this disease in the clinic, and one of the major reasons is that we have a very difficult time getting treatments across the blood-brain barrier and into the tumor. So what is the blood-brain barrier? I've gotten this question a lot, and I think when people first hear about this term, blood-brain barrier, they think of a membrane or a wrapping that might go around the entire brain and keep things from getting in. But it's actually a lot more complicated than that. So within our brain, we have many blood vessels that flow within. And when we get down to the very smallest ones, they're actually wrapped by several layers of cells that create these tight junctions. And these serve a number of roles. For one thing, they help us to get oxygen and nutrients into the brain in a very regulated manner. But unfortunately, in our case, they also keep drugs out. And so in the images that Jeff and I took of the blood-brain barrier, we're actually able to image these very small vessels that are really on the order of what we would call a capillary. And that means that they're so small that's really only allowing 1 or 2 cells to fit through at a given time. And this is where most of the transport actually happens. So if it's playing, you may notice some very kind of small movements. So what I'm showing here is one of the images that's in our panel from the Image Awards. And now we're looking at about a 10-minute long imaging session condensed into a little video here. And what you're looking at on the left in red is a vascular dye that we inject through the veins, similar to how we might give a patient an injection, but it cannot leave the blood vessels. And in green over here on the right, you're actually seeing nanoparticles. And in this case, they're filling the blood vessels, and some of them are getting out past the blood vessels into the brain tissue. And these little dark dots you're seeing are actually shadows of red blood cells. And so in some of these very small vessels, you can see that only 1 red blood cell at a time fits. And this is another example of one of the images from the Image Awards panel. And here you're looking at the blue with this vascular dye. The magenta is actually the nanoparticle signal, and then we've merged them together over here on the right. And in this case, you can see that the nanoparticles are, in this case, a little bit less in the blood vessels, and they kind of appear as a speckled pattern throughout this dark area. Now, it's important to note that you're not actually seeing individual nanoparticles here because those are too small to really see, even at this level of magnification. But what instead we're seeing is cells that have taken up the nanoparticles and kind of make this clumpy speckled appearance. So why are we doing this, and what is the point of taking these images? So at this point, we've tested 3 different nanoparticles, and what we're doing is taking the time-lapse videos that you see here and using computer algorithms to actually see the change in the nanoparticles from inside the blood vessel to inside the brain tissue during the course of the imaging. And this lets us design nanoparticles that are more effectively able to get into the brain and ultimately into brain tumors. And so when I see these images, I do see the beautiful images of the blood-brain barrier, but more than that, I see that we're able to work with a great team of scientists here at the Koch Institute and actually design drugs to go into these nanoparticles. And those are kind of the next steps to really getting this out to patients. So with that, I'd really like to thank you all for your attention. I'd really like to thank Jeff. We've done a lot of work on these images together and spent a lot of time doing the imaging. Could not have been possible without the Swanson Biotechnology Center here at the Koch Institute. Thank my mentor, Paula Hammond, for all of her support. Our technician, Hannah Stafford, who was my surgical assistant throughout all of this, and of course, all the funding sources.
Thank you, Joelle. That was great. You've tempted us with the promise of particles that we can't actually see. So for our next presentation, we're actually going to zoom out a little bit to the micro scale from the nanoscale and take a look at one particular particle with a very particular function.
Thanks, Erica. Good evening, everyone. I'm Ruojie Vanessa Zhang. I was a postdoc from Anna Jaklanic and Bob Langer's group and currently work at Reynolds American. This image was taken during my very last experiment before I moved to my current job. It is my big honor to receive this award and share the story behind this image. This project is one of the studies funded by Gates Foundation that aim at fighting malnutrition for humans. The image showed here is a well-designed microparticle that used for encapsulate micronutrients. The green color is a coating material that used for stabilizing the micronutrients and protecting them from heat degradation, which is a common side effect in cooking. The purple color on the outside is starch particles that used for making the microparticle more flowable, allowing it to be incorporated into foodstuffs easily. The basic idea of this project started with Gates Foundation, including Bill Gates himself, recognized that nearly 2 billion people in the developing world are plagued by micronutrient deficiency, which causes up to 2 million childhood deaths per year. Micronutrient deficiency contributes to many disabilities and diseases, including cognitive and physical disorder, anemia, blindness, birth defects, and impaired growth in children. Food fortification is an effective strategy to increase the daily intake of the stable micronutrients. Unfortunately, many of those findings have not impacted the vast population currently suffering from the micronutrient deficiency because of implementation challenges in targeted countries and technical challenges related to micronutrient stability during the storage and cooking. Target population in developing countries consume food products that often require extensive cooking, which introduce heat, moisture, and oxidation and cause significantly degradation of the fortified nutrients. Therefore, our aim of this project is to develop a microparticle to encapsulate micronutrient in it. When it experiences extensive cooking or storage in hot and humid conditions, the microparticles can keep stable to protect the micronutrients from degradation. Well, when it is ingested, the micronutrients can quickly release from the microparticles in human digestive system. Let's briefly go over how we made this magic happen. We did an in-depth analysis of more than 50 potential polymers and then select BMC as the coating material for micronutrients. BMC is a polymer encapsulant that has historically been used in nutraceutical and pharmaceutical products. And it can form a very compact structure on the surface of the microparticles, providing a moisture barrier and high refractive index surface that can protect the encapsulated micronutrients against heat degradation and UV radiation. BMC also has the properties to quickly dissolve at low pH condition. Therefore, the solubility of the BMC enables the microparticles to rapidly dissolve and release micronutrients in stomach, which has a high acid condition. The exceptional cooking stability of the microparticles can also be seen in the example of the vitamin A. About 50% vitamin A degraded after only 15 minutes under cooking condition, while this process lasts to 5 hours in the case of vitamin A encapsulated in BMC microparticles. We applied this technology into 11 different micronutrients, and all of them can be quickly released in stomach. The human clinical trial has also indicated that the coating material does not significantly hinder the absorption of the micronutrients. This technology can be potentially applied into food applications such as staple food and bouillon, which are widely consumed in target developing countries and have a high chance to experience extensive cooking. We look forward to translating this technology so that all humans have the nutrition they need for a healthy life. I want to thank my advisors, Anna and Bob, for their support and guidance, and all lab members who work on this project. And a special thank you to David from Nanotechnology Materials core facility, helped us to take this image. Last but not the least, I would like to thank Gates Foundation for their funding support. Thanks to all of you for listening tonight.
Thank you, Rogier. That was great. I love the Image Awards because they just provide so much hope about what is possible, both in science and out in the world. All right, so our final presentation this evening is a little bit different than the rest. The 10th image in our 2021 Image Awards is an encore from 2011. A miniature mesh appeared in the very, very first Image Awards exhibition with a very simple and intriguing question. If you link viruses together or encourage them to do it, what are the structural properties of that mesh that they create? And so the PI from that project, Angela Belcher, is going to talk to us about what they've done in the 10 years since this image first appeared in our galleries.
Thank you. Can you see my screen?
You can see your screen. We can't see you.
Yeah, okay, let me go back out for 1 second. Oh, it says the host has stopped my video. There, okay. Great. Well, thank you so much. I'm excited to be part of this kind of flashback image. So I'm sitting in front of this image of a beautiful network or phage of M13 bacteriophage. And the image that we originally submitted in I think it was the first KI Image Awards was viruses that had been genetically altered, M13 bacteriophage that had been genetically altered to grow battery electrodes. And this image was taken by a former graduate student of mine, Colonel John Burpo, who's now a professor at West Point. And so what this image showed was that we could take these M13 bacteriophage, which are beautiful, long, thin viruses shown right here that have a DNA sequence that codes for the proteins that make them up. We could genetically program them to grow a whole bunch of materials that we're interested in. In that case, it was for battery electrodes. Over the last 10 years since this time, I have not lost my love for the structure of M13 bacteriophage. This is a picture here of the M13 bacteriophage showing the kinds of things that you can build out of it. And this is my great collaborator, Paula Hammond, and she and I worked together to build phage-based solar cells and batteries. We also showed that you could use these same structures that were in the image from 2011 to build catalysts, drop-in fuel replacements, building supplies, and we moved deeper into understanding ovarian cancer using these same bacteriophages, as well as artificial photosynthesis as well. This is a movie of where we went in batteries. This is a virus that has been engineered on its beautiful little protein coat to be able to nucleate in solution out of water, manganese oxide solution with a small amount of palladium. What we showed is that the virus template made these really great nanostructures that were high surface area and very reactive as a catalyst electrode in a lithium oxygen battery application. And we showed that through this genetic engineering over time, that we could get record-breaking lithium oxygen battery electrodes. And we thought that one day these could be used for electric vehicles to increase the time between charging. And so here it is functioning in a device with the idea that it could be eventually used in transportation. Since then, we have also used these same kinds of structures to absorb small gas molecules, including CO2, and we've used them to take CO2 and convert them into other more useful products, including energy storage for batteries or smaller molecules. But we're in the Koch Institute, we're in the KI, and we wanted to use them for applications in cancer. And the particular kind of cancer we wanted to start with was ovarian cancer. And the reason we wanted to look at ovarian cancer was because if you catch ovarian cancer very early, you have a good 5-year survival rate, but most of the time it's not caught early. And at distance, you have a 29% survival rate. So we thought, what can we do with our expertise in phage and nano to work in this area? So what we did, and this is in collaboration with Sangeeta, in collaboration with Paula Hammond on certain ovarian projects. And what we did was we engineered these viruses to find tumors and to carry with them a structure called a carbon nanotube that's fluorescent in wavelengths that are in what's called second window near IR, so imaging above 1,000 nanometers. This was really important because it allowed us to image inside of the mouse or inside other organisms without having to open them up, and so looking deeply. And so this image shows basically pre-injection. This is a mouse that has a high burden of ovarian tumors. This is what it looks like pre-surgery, where you can see a high density of tumors. A surgeon goes in and doesn't look at our map of where we think the tumors are and just does surgery and leaves a lot of tumors behind. And then if we give him the map, he was able to go in and remove more tumors. So we're using these viruses now as vehicles for imaging agents. And what we found is that we could find tumors that are much, much smaller, tumors that are on the millimeter length scale with these types of virus-based delivery systems. We went on to build a real-time surgical guided imaging system with Lincoln Labs, MIT Lincoln Labs and Mass General, where we could help surgeons find the tumors pre-surgery and then help them during surgery to remove very small tumors. These are tumors that are millimeter-sized tumors. And by doing that, we were able to have a 40% increase in survival rate of the mice in this example. We're now working on sending these materials for testing at NIH, and we're working on a human scale instrument for image-guided ovarian cancer surgery. And this is showing that we can find very small tumors that are even less than a millimeter in size. something that's new, that's also pretty cool, that's based on the same structure, is that we're able to make really high surface area carbon viruses that can neutralize chemical warfare agents like sarin and soman, and they can neutralize toxic industrial chemicals. And so we worked with the Army to build masks out of these that could be used by first responders in difficult situations to neutralize chemicals that they would come in contact, including very, very deadly chemicals. And this graph here shows what our material does. Gives a long time before you actually, the chemicals break through. We said, well, wow, if you can do that, maybe we could make masks for smog and pollution and wildfires based on the same kinds of structure, which we did. And right before the pandemic, we started working on making masks that could prevent viruses from filtering in. And so we repurposed the same structures we use for batteries and for chemical warfare agents, and we used them to make masks that could not only prevent very small viruses that are smaller than the SARS-CoV-2 from entering, but could also neutralize them. So this data here shows that these two formulations of viruses we engineered to grow carbon-based structures neutralizes viruses that were normally 1 × 10⁻11 to 10 viruses that are still able to replicate. So right now we're very interested in transitioning this for future kinds of viruses, bacteria and viruses that could be helpful for doctors, first responders. And so with that, that tells you kind of the evolution of where we're going. We think the M13 virus is amazing material that can be genetically engineered and altered for many different kinds of applications. And the image that we showed 10 years ago is being tested in drone wings to increase the flight time, battery flight time in drones, to all kinds of other batteries. And that technology has gone on to look at cancer and gone on to look at also ways of preventing the spread of viruses and a way of neutralizing chemical warfare agents, which could be used for first responders, or eventually one day used for people that are in situations where in other countries where they may come in contact with very toxic chemicals. With that, I'd like to thank you for including our flashback material. And thank you.
Thank you. And thanks for all the work you're doing. I hope that in our 2031 exhibition, we'll be able to revisit many of these projects that we have seen over the years, and with particularly in tonight's event. I think you've all opened up our minds to a wide range of possibilities and the role that images can play in helping us understand these things. So we've come to the end. So I encourage everyone to be on the lookout for our events calendar for our anniversary year. I'm going to leave things open a little bit longer in case anyone has any last questions that they want to do. I have a video that I'll play as we say goodbye for the evening. Thank you for celebrating the Image Awards with us. Thank you for logging on, and thank you for all the work and all the support you're doing of science.